So next we want to talk about genetic variation. So what's the significance of genetic variation? Why do we care about genetic variation? Well, genetic variation is how species are able to survive and adapt to their environment. And I know we haven't really talked heavily about natural selection, but if you remember early on in chapter 1, when we first started talking about it, we were talking about how leaves in the forest may be, deer in the forest may have a preference for smooth leaves versus hairy leaves. If we have genetic variation in the population, if we have a population of trees where some produce smooth leaves, some produce hairy leaves, some are really tall, some are really short, the greater the variation in the population, the greater the chance that the population is going to survive when natural selection starts whittling down characteristics that are most beneficial to that particular environment. So genetic variation is huge and hugely important. It's absolutely essential for a species to be able to evolve and adapt in a constantly changing environment. Asexual reproduction, or asexually reproducing organisms, those that can reproduce by themselves, like bacteria, they have to rely on random genetic mutations in order to generate the genetic variation within their lineage. So the only way that they can become immune or resistant to particular antibiotics is if there's just some random mutation that just so happens to be present that allows them to survive in that kind of environment. Meiosis meiosis in sexually reproducing organisms can greatly speed up the process of introducing genetic variation. And it does this in one of two ways. One would be crossing over events between homologous chromosomes, and two would be independent assortment. We're going to dive into both of these. Crossing over events. Now, crossing over events, these are bizarre and really cool. Crossing over is an exchange of genetic material between non-sister chromatids during meiosis. So this is where things get a little bit wonky. What happens is in our homologous chromosomes, remember when we talked about how our homologous chromosomes are going to synapse during prophase 1? At the synapse, the nucleoprotein lattice called the synaptimal complex appears between the two homologs. So in other words, not only do they pair up, but then they start building a structure in between them. During this time, while the homologs are together, they start swapping DNA. Yeah, we're going to start swapping DNA of non-sister chromosomes. This allows for a crossing over event or exchange of genetic material to occur. And then, once they've all swapped their DNA, the homologs are going to separate, and they're going to be distributed into different daughter cells. So here's just an example of what that looks like. We started off with 2 homologous chromosomes, but what's going to happen when we line up for the synaptimal complex, we're going to start swapping DNA. We're going to start, maybe we'll have a little bit of dad's DNA get swapped with a little bit of mom's DNA, a little bit of mom's DNA get swapped with a little bit of dad's DNA. Some of those chromosomes, or some of those sister chromatids might not swap DNA at all. Some of these crossing over events can happen multiple times. Some will happen hardly ever. So when these separate back out and we have our daughter chromosomes, we're going to end up with 4 completely distinct eggs, or sperm, 4 completely distinct gametes that are going to have absolutely nothing identical to the parent or to the parent DNA. So we're going to see complete randomization of this genetic material during these crossing over events. So that's one way we get genetic variation. Another way that we get genetic variation would be through independent assortment. Independent assortment is also pretty cool. Independent assortment of the homologous chromosomes means that when those homologous chromosomes line up down the center of the cell, they could line up in any which way. We have no idea how they're going to line up. So you could line up all your chromosomes with all 23 of your mom's chromosomes on the left side and all 23 chromosomes of your dad or your mom on 23 chromosomes of your mom on the left side, 23 chromosomes of your dad on the right side. When this egg splits, you're going to see, or when this gamete, when this when this parent cell splits, you're going to see that one daughter cell is going to look an awful lot like your mom, and the other daughter cell is going to look an awful lot like your dad. But that's just one possibility. You could also have, two-thirds of your mom's chromosomes on the right and two-thirds of your dad's chromosome on the left. You could have an every other. You could have, there's any sort of combination. So if you had only 3 different chromosomes in your body, and keep in mind, you have a lot more than that. You've got 23 chromosomes. But if you had only 3 different chromosomes in your body, you would have gametes that could have one of 8 possible genetic combinations. So you get 8 different variations from having only 3 chromosomes. Plus, you got to keep in mind that each one of those homologous pairs could have undergone those crossing over events. Fertilization is another way that we can get even more genetic variation. So that's the union of the male and female gametes. You're going to get chromosomes donated by the parents that are going to be combined together. So in humans, and I'm not going to ask you to memorize any of these numbers, I'm just throwing this out there. When you look at the number of combinations that are possible between a male and a female during that fertilization and the arrangement of the chromosomes, there is, hundreds, thousands, millions, billions, trillions. There are 70 different trillion possible combinations. That means that if your parents had 70 trillion offspring, you are 1 in 70 trillion. The odds that another offspring is going to be exactly like you are 1 in 70 trillion. So if anybody ever accuses you of thinking that you're like, oh, some sort of special little snowflake, you kind of are. You kind of are. You're a 1 in 70 trillion chance of being the genetic person that you are. This is why there has been so much pressure, or not pressure, but movement in the direction of DNA analysis, DNA fingerprinting analysis, as opposed to just looking at fingerprinting or blood typing, because the possibility that another individual out there has your exact same genetic variation is 1 in 70 trillion. It's just not likely at all. And that's if crossing over, it doesn't exist. But if crossing over events occur only once in every single chromosome, that number goes from 70 trillion to an insanely large impossible number that I don't even know. Wait, we've got hundreds, thousands, millions, billions, trillions. I don't even know after that. What's a number that is 1, 2, 3, 4, 500 times larger than a trillion? So I don't know, it's massively huge, massively huge. And that's if crossing over only occurs once. However, crossing over events can occur several times for every single chromosome. You're a freaking 1 in a million snowflake, not even a 1 in a million. You're a 1 in an insanely ginormous number that I can't even mentally comprehend. So there you go. If anybody ever accuses you of thinking that you're some sort of special little snowflake, you should just point out that you kind of are. You kind of are. So anyway, I just wanted to point that out because I think that's absolutely fascinating, and I think it's also very important to understand why we put so much pressure on DNA analysis. Because if there's nothing wrong, as long as that DNA sample has not been compromised, as long as there's no, as long as that DNA sample has been handled correctly, you can't fake. I guess you can't mistake a DNA sample. If the science is done correctly, if everything has been handled correctly, it's not like there could be a mistake of another person on the planet who has the exact same genetic information as you. Unless you have an identical twin, and we're going to talk about that a little bit later when we get into cloning. But just know for now, as long as you do not have a genetically identical twin, if you were not born with a genetically identical twin, you are a 1 in insanely billion chance of happening, just in terms of your DNA alone. All right, off my soapbox. On to the significance of genetic variation. As I mentioned before, asexual reproduction produces genetic clones. Bacteria, when you see a whole colony of bacteria, every single bacteria in there is a clone of the original bacteria who started that colony. Asexual reproduction is advantageous when the environment is stable. There are positives to having asexual reproduction if the environment's not going to change. if you know the environment is not going to change, if you know the temperature is not going to change, you live inside of this human gut, you live in their your bacterial colony, and you live in the human gut, you know the environment's not going to change, the pH is not going to change, not unless there's something truly terribly wrong. Then it is beneficial to just keep cloning yourself. You know, why fix what ain't broken? It's a waste of time to put energy and resources to genetic variation when you've already got, you're already optimized for those conditions. If, however, the environment is unstable, sexual reproduction is the way to go. It causes genetic recombinations among members of a population. In a changing environment, the genetic variability introduced by sexual reproduction can be extremely advantageous. Some offspring might have better chances of survival than others. If you live in really cold, frozen tundra, it might be really good that, you know, one or two of your offspring have a thicker fur. If you're a little hutch of rabbits, it might be pretty good if some of your rabbits have a bright white coat. I mean, it might not be advantageous for some of them to have a really dark coat or have a black coat or have a really thin coat. That might not be so good. But if you get one or two freaks in there that have a really thick, furry white coat that helps them hide in the snow and survive, there is a good chance that they are going to thrive in that environment. So, sexual reproduction is fantastic for the introduction of genetic variation, and that's really excellent in an unstable environment.